The advent of genomics has revolutionized our understanding of living systems, providing a comprehensive view of the genetic blueprint that underpins the complexity of life. However, the vast information encoded within genomes does not operate in isolation. Rather, it interacts through intricate networks of genes, proteins, metabolites, and environmental factors. Systems biology emerged as a response to the limitations of reductionist approaches, seeking to integrate data across multiple biological layers to elucidate the dynamic behavior of whole systems. The integration of genomics and systems biology represents a paradigm shift in life sciences, enabling researchers to move beyond cataloging genetic elements to deciphering how these elements interact to give rise to phenotypes, disease states, and responses to environmental cues. Genomics at its core involves the sequencing, annotation, and analysis of entire genomes. Since the completion of the Human Genome Project, advances in high-throughput sequencing technologies have dramatically reduced the cost and time required to sequence DNA, making it feasible to analyze the genomes of thousands of species as well as the genetic variation within populations. This deluge of genomic data has provided unprecedented insights into gene content, structure, function, and evolution. Yet, possessing the sequence of a genome is only the first step toward understanding the functional output of a biological system. Genes do not function in isolation. They are regulated by complex networks that include transcriptional, post-transcriptional, translational, and post-translational mechanisms. Moreover, the activity of gene products is influenced by interactions with other molecules and the environment, necessitating holistic approaches for meaningful interpretation. Systems biology addresses these challenges by adopting a holistic, integrative approach to biological research. Rather than focusing on individual genes or proteins, systems biology seeks to understand how components interact to form networks and pathways that govern cellular and organismal behavior. This approach relies heavily on mathematical modeling, computational simulations, and the integration of diverse datasets, including genomics, transcriptomics, proteomics, metabolomics, and phenomics. By constructing and analyzing models of biological systems, researchers can generate hypotheses, predict system behavior, and identify key regulatory nodes that may serve as therapeutic targets or biomarkers. The integration of genomics and systems biology has catalyzed numerous breakthroughs in our understanding of complex traits and diseases. For example, genome-wide association studies, GWAS, have identified thousands of genetic variants associated with common diseases such as diabetes, cancer, and cardiovascular disorders. However, many of these variants reside in non-coding regions of the genome, making it difficult to infer their functional consequences. Systems biology provides the tools to contextualize these findings by mapping variants onto regulatory networks, identifying the genes and pathways they influence, and exploring how perturbations in these networks contribute to disease phenotypes. This integrative approach has led to the discovery of novel disease mechanisms and the identification of potential drug targets. One of the most striking examples of this integration is in cancer research. Cancer is not a disease of single genes, but a multifaceted process involving alterations in multiple molecular pathways. Genomic analyses have revealed the extensive heterogeneity of cancer genomes, with each tumor harboring its own unique constellation of mutations, copy number variations, and structural rearrangements. Systems biology approaches have been instrumental in interpreting this complexity by constructing network models that capture the interplay between oncogenes, tumor suppressors, signaling pathways, and the tumor microenvironment. These models have enabled the identification of driver mutations, prediction of drug responses, and the development of personalized medicine strategies tailored to the molecular profile of individual tumors. Another area where genomics and systems biology converge is in the study of developmental biology and stem cell differentiation, the process by which a single fertilized egg gives rise to the multitude of cell types in an organism is governed by tightly regulated gene expression programs and signaling networks. High-throughput sequencing technologies have made it possible to profile gene expression at single-cell resolution, revealing the diversity of cell states and trajectories during development. Systems biology methods, such as network inference and dynamical modeling, have been used to reconstruct the gene regulatory circuits that drive cell fate decisions. This integrative approach has deepened our understanding of developmental processes and holds promise for regenerative medicine, where directing the differentiation of stem cells into specific lineages is a major goal. Microbial genomics and systems biology have also joined forces to advance our understanding of microbial communities or microbiomes, which play critical roles in health, agriculture, and the environment. Metagenomic sequencing allows researchers to characterize the genetic composition of entire microbial communities without the need for cultivation. However, to understand how these communities function and respond to perturbations, it is necessary to integrate genomic data with information about metabolic pathways, ecological interactions, and environmental factors. Systems biology approaches, such as metabolic modeling and community network analysis, have been used to predict the functional capabilities of microbiomes, identify keystone species, and design interventions to modulate community composition for beneficial outcomes. The integration of genomics and systems biology has been greatly facilitated by advances in computational tools and data science. The sheer volume and complexity of omics data requires sophisticated algorithms for data integration, normalization, and analysis. Machine learning and artificial intelligence are increasingly being employed to extract patterns from multidimensional datasets, identify novel associations, and predict system behavior. Network analysis tools enable the visualization and interrogation of complex interactions, while dynamical modeling provides insights into the temporal evolution of biological systems. The development of public databases and data sharing platforms has further accelerated progress by enabling researchers to access, compare, and integrate datasets from diverse sources. Despite these advances, several challenges remain in the integration of genomics and systems biology. One major challenge is the accurate annotation of genomes, particularly in non-model organisms, where functional information is limited. Many genes remain uncharacterized, and the regulatory elements that control gene expression are often poorly defined. Experimental validation of predicted interactions and network models is essential, but can be laborious and resource-intensive. Furthermore, biological systems are inherently dynamic and context-dependent, with gene regulatory networks reconfiguring in response to developmental cues, environmental stimuli, and disease states. Capturing this dynamism requires time-resolved data and sophisticated modeling approaches capable of accommodating feedback loops, stochasticity, and nonlinear interactions. Another challenge lies in the integration of heterogeneous data types. Genomics provides information on DNA sequence variation, while transcriptomics measures gene expression, proteomics quantifies protein abundance, and metabolomics profiles small molecule metabolites. Each data type has its own technical biases, noise characteristics, and temporal resolution. Effective integration requires careful experimental design, robust statistical methods, and the development of standards for data representation and sharing. Interdisciplinary collaboration between biologists, computer scientists, statisticians, and engineers is critical to surmount these challenges and realize the full potential of integrative approaches. Ethical, legal, and social implications, ELSI, also arise from the integration of genomics and systems biology. The ability to generate and interpret vast amounts of personal genetic and molecular data raises concerns about privacy, data security, informed consent, and the potential for misuse of information. As integrative approaches become increasingly central to precision medicine and population health initiatives, it is imperative to establish frameworks that protect individual rights while enabling scientific progress. Public engagement, transparent communication, and the development of ethical guidelines will be essential to navigate these complex issues. Looking ahead, the integration of genomics and systems biology is poised to transform biomedical research, clinical practice, and biotechnology. In medicine, the shift toward precision health will be underpinned by the ability to model individual patients as dynamic systems, integrating genomic, molecular, physiological, and environmental data to predict disease risk, tailor interventions, and monitor responses in real time. In agriculture, integrative approaches will inform the development of crops and livestock with enhanced productivity, resilience, and nutritional value by unraveling the genetic and network basis of complex traits. In environmental science, systems-level understanding of microbial communities and ecosystems will guide strategies for bioremediation, conservation, and sustainable resource management. Emerging technologies are set to further accelerate the convergence of genomics and systems biology. Single-cell multi-omics, which simultaneously profiles multiple molecular layers within individual cells, promises to reveal unprecedented insights into cellular heterogeneity and network dynamics. Spatial transcriptomics and imaging mass spectrometry allow researchers to map the spatial organization of molecules within tissues, linking genotype, molecular phenotype, and tissue architecture. CRISPR-based genome editing enables precise perturbation of genetic elements, facilitating the experimental validation of network models and causal inference. Synthetic biology, which combines principles from engineering, genomics, and systems biology, is opening new frontiers in the rational design of biological systems with desired properties. Education and training will play a crucial role in fostering the next generation of scientists capable of navigating the interdisciplinary landscape of genomics and systems biology. Curricula that blend biology, mathematics, computer science, and engineering will equip students with the skills needed to analyze complex data sets, construct and interpret models, and translate discoveries into practical applications. Collaborative research environments that bring together experts from diverse fields will foster innovation and accelerate the pace of discovery. In conclusion, the integration of genomics and systems biology represents a transformative approach to understanding the complexity of living systems. By moving beyond the study of individual genes to embrace the networks and pathways that govern biological function, researchers are uncovering the principles that underlie development, disease, and adaptation. The synergy between high-throughput genomics and systems-level modeling is unlocking new avenues for diagnosis, therapy, and biotechnology, with profound implications for human health, agriculture, and the environment. While challenges remain, continued advances in technology, computation, and interdisciplinary collaboration promise to propel the field forward, ushering in an era of truly integrative biology. As we deepen our understanding of the genome, and its dynamic interactions, we move ever closer to realizing the vision of predictive, preventive, and personalized approaches to health and disease, a testament to the power of integration in the life sciences.